introduce Dr. Pat Wyse to the podium. Dr. Wyse is a clinical associate professor in the departments of surgery, that's a division of ophthalmology, and the medical genetics department at the University of Calgary. He's trained both as a vision researcher and a clinical ophthalmologist. He provided expert care for people with retinitis pigmentosa and other inherited retinal diseases. Now semi-retired, Dr. Wyse continues to nurture an intense curiosity about the causes and treatments of eye disease. So without any further ado, please welcome Dr. Pat Wyse.
Thank you very much for that introduction. In life, as we go along, we learn lessons, and one of the lessons that I learned early on, if I was going to go for a long walk, I didn't buy new shoes and wear them because I was going to get blisters, and that didn't transfer to my eyes. I just got new glasses yesterday, and I'm having a heck of a time seeing this screen. So we'll have some fun with that as we go along. The topic that I'm going to present today is it's complicated. So my challenge is to simplify it, because the concepts that are involved are simple, and they're concepts that we can all understand if they're verbalized in a way that relates to our common everyday experience, and they can be. So if you don't understand it, it means I've done a bad job at the end. Now, throughout the talk, I'm happy to be interrupted and for questions to be asked. Now, I've labeled this talk in part, well, I'll give the whole title, Macular Degeneration, Advances in Stem Cell Therapy, Hard Work, Hype, Hubris, and Hope. Now, there's been an enormous amount of hard work that has gone into this, and it is hyped up. And you don't have to go very far in the newspaper and the media to be hearing about the benefits of stem cell therapy and new work that's going on in stem cell therapy. And we had an incident in Calgary here about 3 years ago. A young boy was born with underdevelopment of his optic nerve, and the CBC got behind this, and there was a big media push to raise money so that this young boy could go to China to get stem cell therapy. And I was absolutely appalled by it because there was no science behind it. And what they were going to do was to inject stem cells into this boy's spinal space, and hopefully that the stem cells were going to repair his visual problem. And that's not going to happen. So that's part of the sales oil, the snake oil salesman pitch that you might hear. And as medical consumers, you've got to be vigilant. And what I hope to do in this talk is to give you some background that you can provide judgment to those things that you're hearing. I put hubris in here because so much of what has gone on in this topic has there's so much pressure in it, and people being people and being highly motivated to try to exceed and excel have sometimes stepped beyond the boundary of what their data was really showing. And I was watching very closely because there was such an incident in Japan, and one of the papers that I'm going to be presenting, one of the clinical trials I'm going to be presenting, came out of a big research center in Japan. And the researchers in that center had indicated that they could create stem cells from ordinary skin cells by simply heating them or putting them in an acid solution. And that really excited the world, because that meant, we might not have to use embryonic stem cells, and I'll talk about that later. But it turned out that data was all bogus. It was the hubris that was there. It was the human drive to want to achieve and excel, and the boundaries were crossed. And it turned out that data those papers were withdrawn, and the head of that lab perhaps a reflection of the society that it came out of, committed suicide in August. And so that is part of the hubris, and it's part of the drive that's behind. There's a lot of very, very motivated people that are working on this around the world. But with all of that in there, I think the underlying statement has to be one of hope, and I hope to be able to get that across to you by the end of the talk today. So really the question is, and I don't have Dr. Amin Karania, if you listened to that talk where Dr. Karani was asking Dr. Williams questions, and I'm going to have to ask myself questions, and I don't have a split personality, so I will ask the question and answer it as well. And so can stem cell therapy be used to regenerate a degenerated retina? That's the fundamental question. So right now the answer to that question is no, but clinical trials are underway which are exploring that possibility. So we will delve into those studies, but before we do so, we need to review some background information. So let's do some exploring together. First of all, in the talks that I've heard today, I've not really heard anybody say what the retina is, and that may have been said, but I didn't attend all of the sessions. And what the retina is the nerve tissue that lines the inside of the back of the eye. It's derived from brain tissue, and it has a cell makeup that allows it to receive light from the external environment and transform that light into cell signaling, which can be sent back to the brain. And clinically, when an ophthalmologist looks in your eye, he's looking at the nerve tissue in the back of the eye, he's looking at the optic nerve. And for those of you that are sighted in the picture in front of you on your right-hand side, there's a little round structure that's sort of light-colored, and that's the optic nerve, and it carries 1 million nerve fibers from each eye back to the brain. And the actual size of that structure in the eye is about 1.5 millimeters in diameter. So you've got 1 million nerve fibers that are going through that tiny little space and carrying information back to the brain. What we're interested in from the stem cell therapy part of it is not what's going back to the brain, but what's actually receiving the light, and it's the photoreceptor cells, the rods and the cones that you will have heard about today, and the cells that they're immediately in contact with, which are the pigment epithelial cells. And those are the two cells that we want to talk about, the two groups of cells that we want to talk about today. So within the retina is made up of a number of different cells. And in terms of an organizational structure, we have the cells that detect light, the rods and cones. When they get their signal, then they speak to the next cell in line. And it's sort of like the game that we all played as kids, where you whispered the message around the table to see what the answer was at the end. And that's essentially what's happening with this, where the rods and cones are talking to the next order cell, which are called bipolar cells, which then talk to the next order cells, which are called ganglion cells, and the ganglion cells send the message to the brain. And then there are little cells that are in there that are trying to interpret it and sort of muddle that up a little bit, and they fine-tune the sequence, and those are called bipolar cells and horizontal cells. We don't need to know that, but the point of it is the retina itself has quite a number of different cells in it that are all working together and are integrated. So from the point of view, I should have said in the previous slide, from the point of view of the stem cell therapy, the stem cell therapy that is going on right now, the clinical trials that are going on right now, are not regenerating the neural part of the retina. So the bipolar cells, the ganglion cells, the amacrine cells, or the horizontal cells, they're targeted at generating a relatively simple layer, the pigment epithelium. But as an offshoot, they're also producing photoreceptor cells, and I'll talk about that. But the stem cell therapies that are underway, the trials that are actively underway right now, are designed to regenerate the pigment epithelium. So where did the retina and the retinal pigment epithelium come from? You know, they're sitting in the back of the eye in the adult, but they weren't always that way. And they're actually derived from, embryologically, from an outpouching of the brain. The brain in an early development, and as part of our development, we all went through it. We might look at some of our family members and say, "What a worm!" but the truth of it is, we all went through that stage. We went through a stage in our development, an embryologic development, that we kind of looked like a worm. And in that early worm-like stage, there starts to be some development of the head of the organism, us in that case, and the inside layer of that is a part of the developing brain, and it gets little pouches that come out the side of it. And for those of you who are sighted, we'll see that there's this ellipse, and inside the ellipse there's a darker zone with little pouches that come out on either side. And those little outer pouches are what are going to give rise to the eyes. And they move out, come in contact with what will become the skin and is what's called the epidermis, and they start to talk to each other. And the skin says to that brain that's coming out, I want you to make a little bulge in. And they're talking like that. But they're not talking in the language that we're talking in. They're talking in chemical language. And the challenge is to understand what that language is. So I've used the word stem cells a bunch of times. And how many in here know what stem cells are or have any idea of them? So we got a number of hands that went up. So stem cells are unspecialized cells in the body. And they have a number of features. They can duplicate themselves. So if you take a stem cell, put it in the right kind of a medium, it'll make 2 stem cells. And it'll make 4 stem cells. And it'll make 8 stem cells. And it'll keep on doing that ad infinitum. Whereas pigment epithelial cells and photoreceptor cells and the ganglion cells and the horizontal cells can't do that. So stem cells have a special characteristic of being able to reproduce themselves. But they can also be transformed into specialized cells, like photoreceptor cells and pigment epithelial cells. So they have that capacity. And so, I got some words here. I'm going to read it because I worked hard at putting this together in the right way. So there is an orchestrated transformation of the stem cells. So you think of the conductor, okay, and the conductor is coordinating the whole orchestra. And so there's an orchestrated transformation of the stem cells in development, which in a stepwise fashion defines subsets of genes. So we've got this great big genome. We've got all these genes, and we've got to take a fertilized egg and take those genes and say, well, we're going to put a subset of you in to make a retinal cell, but we're going to take another subset of you and make a liver cell. And how the heck does the body know how to do that? And so that comes through this orchestrated definition of different subsets of genes that are expressed in succeeding generations of stem cells, and which ultimately lead to fully differentiated and specialized cells which cannot duplicate. So the gift of duplication is conferred upon stem cells. The control of mature cells is they cannot divide. And that's a protection against cancer. And I'm going to talk about that as we go through it. So where do the stem cells come from? Well, we're all here because of it. And that is, there's a sperm found an egg and turned that into what we call a zygote. And the zygote is the fertilized egg, and it's what's called a totipotent stem cell. That means it can become anything that a human or whatever the species would be. But it means that it can become the embryo, it can become part of the placenta, it can become the lining of the uterus. It can do all of those things, and that's in the totipotent stem cell. And it has the capacity through the interplay of its internal genetic information. So it's got all those genes in there, and its own internal makeup. So each egg has its own internal chemical makeup, which starts to define what's going to happen with the daughter cells. And the external environment to form through a cascade of cellular division and cellular differentiation of all the cells into the adult organism. And I'm going to try and step you through that. Are there any questions at this point in time? No. This is heavy stuff, but it's... Yeah, well, then you be a good judge for me there. Okay, there's a cascade of different stem cells, and they go from the totipotent cells, which are the eggs, into what are called embryonal stem cells, germ cell layers, which are the layers in the skin, the layers in the muscles, and the layers on the inside of us. And then it goes down through a cascade until you get to the terminal cells, which are what are the nuts and bolts of making us work. So here we have a fertilized egg, which divides in two to form two cells, which is the two-cell embryo, and then it divides into 4 cells, into 4-cell embryo, and then an 8-cell embryo. And one of these studies I'm going to tell you about today, done by a company in the States called Advanced Cell Technologies, is isolating one of the cells from the 8-cell stage and multiplying that up in tissue culture to form thousands and millions and potentially billions of copies of that, which stem cells can do. Now, they've got around this because in the States you can't do stem cell research because you're killing the embryo. Their logic is they're only taking 1 cell, so they're not killing the embryo. So I said, well, what have you done with the other 7 cells? Did you freeze those away? No, we know what happened. They went into the garbage can. No, that's what's happened with it in reality. But they've got around it in a sort of that legalistic way by taking that 1 cell. Now, that 8-cell stage then forms a little ball, which is called the blastocyst, a ring of cells, and then there's a little nest of cells inside of that, which is called the inner cell mass. And the inner cell mass is what gives rise to you and to me and all of us in this room. We've all been through that stage. And it's those cells then that become the general germ layers of the body, called ectoderm, mesoderm, endoderm, and germ cells, and those are the ones that give rise to the rest of the body. So stem cells can make stem cells of the same type. They can give stem cells that are more differentiated. They can give rise to mature adult cells, but they can also give rise to cancer cells. And this is one of the things that we have to be very aware of with the cells. So how do cells communicate? And I see a raised eyebrows here. But it's simpler than that, because the cells communicate by chemical signals. They talk to each other with chemicals. They release chemicals, and they'll respond to that. They also respond to what's called extracellular matrix, their surrounding tissue, the substrate they live in. And I think of the extracellular matrix as the biologic marionette. You know what a marionette is. It's the puppeteer, that's moving the puppets around with the strings. And the connective tissues are like strings that are pulling on those cells and helping to tell them what to do and what to become. So we've got cell signaling. We've got the marionette. And then there's another thing called the primary cilia, which every single cell has. And they were thought to be of no significance at all until about 10 years, and it realized that they're central to development. They're central to cell signaling, and they're certainly central to vision, and they're biologic antennae. And where do those biologic antennae exist? Well, they're our photoreceptor cells. They're derived from those cells. And so are the cells in our nose. All of our distant senses are these primary ciliated cells. And so they're really important, and they're also very important in our development. The interesting thing about primary cilia is they've got little bodies at the base of them. It's like a root, and that root is required for cell division. If that root is locked up in the primary cilia, the cells can't divide. So in order for the cell divide, you've got to get rid of your primary cilia. You take those little structures that are called centrioles, and they go into the cell, and they pull the chromosomes apart to make the 2 daughter cells. So as cells divide, the primary cilia regresses and comes back. And the thing with cancer cells is they've largely lost their primary cilia. So a lot of the control of cancer is through primary cilia. So how do cells communicate? Well, they also communicate through RNA. Now, up until 10 years ago, and you've probably all read it, that most of the DNA in our cells is junk DNA. 4% of the DNA is transcribed into working proteins. And the rest of it was just thought to be vestigial material. Well, it turns out that's not the case. There's very little of it is vestigial material, and the vast majority of what's not coded is actually pulling strings behind the scenes to tell genes what to do. And so it wasn't junk DNA, it was lack of understanding. So here in our genome, up until 10 years ago, we didn't know that the most of the genome was doing something. We didn't know anything about ciliated cells, so how the heck could we cure something as complicated as macular degeneration if we didn't know all of those fundamentals? So we're learning that, and that's where the hard work is coming in. So what happens within a cell in response to those different communication codes? And so I thought about this in a way. All of us have heard of the Rosetta Stone, right? Rosetta Stone, found in Egypt by the French army in 1795. And it had on it ancient Greek, it had on it hieroglyphics, and had on another of the older Egyptian languages. But with hard work and decipherment, they were able to figure out what the hieroglyphics meant by translating it from the other languages. And similarly, you've all heard in the last couple of years about the Higgs boson and the Large Hadron Collider over in Europe, where they've the mathematical abstraction of what reality is. They were able to create a fairly simple model, and by looking at that model and deciphering the code, they understood what's going on in the physical world better. So what's that got to do with genetics, and what that got to do with macular degeneration? Well, it turns out that there are codes that tell cells what to become. And there's a group of what are called patterning genes that tell those early embryonic stem cells what to become. It turns off sets of genes and turns on sets of genes. We're okay. The rest will go fine.
I want to make sure that we have time for questions, and we only have 15 minutes.
We're gonna be fine.
Okay.
The coding genes, and they're called homeobox genes, are very ancient. They're probably 750 million years old, and there are 39 of them in the human. And the bulk of those, the predominance of them, involve development of the face and the head, and very few of them for the limbs. But the thing that's interesting about those genes is exactly those same genes make up a fruit fly. So those exact same genes are in there. It's just we have 4 sets of them, whereas the fruit fly only has 1 set of them. And that's part of the complexity of mammals. So we've got 20,000 genes. Of those 20,000 genes, 2,600 of them are telling the other genes what to do. And so there's only the mortar and the bricks are the difference there. There's only about 3/4 of the genes are the mortar and the bricks. The other ones are instructional genes. So one of the things about transcription factors, and I want to just give one example, it's one called PAX6, and it's a master control gene for eye development. And you can take a PAX6 gene from a mouse and put it into a fly, and it'll induce those cells in the fly to make a fly eye. You know, it doesn't turn around, and you can do it the other way. You can take the PAX6 gene from the fruit fly and put it in the mouse, and it'll induce a mouse eye. And so that system has been there for 750 million years. Now, what we don't see is we don't see taking a fruit fly gene, PAX6, putting it in a human and getting a fruit fly. We don't see it. We don't see that, you know, because the genes it's turning on are human genes. They're mammalian genes. So this is a concept I want to get across. We've got 20,000 protein coding genes. So if we think of development as a board game, we've all played board games. So if in the pieces of the board game we've got 20,000 protein coding genes, and we've got about 20,000 regulatory RNA, so we've got maybe 40,000 pieces in that game. Okay, well, is that complicated or not? Well, let's look at this. If we look at the game of chess, chess has 6 different pieces versus 40,000. Chess is played on a board that's confined to 8 by 8 squares, 64 squares. So we've got way more biologic potential than there is a game like chess. And even with chess, as relatively simple as it is, there are more potential games in chess than there are atoms in the universe. So it gives you some appreciation of the potential complexity of the biologic system. So where does this get us from the point of view of stem cell treatment? There are 2 trials that are underway. One is a trial by Advanced Cell Technologies out of Marlborough, Massachusetts, and they're testing for, they're trying to treat Stargardt's disease, and they're trying to treat dry macular degeneration, and they're using isolated embryonic stem cells. So that cell that I showed you where they took that one cell off, that's what they've done. So they're using an embryonic stem cell, and they are developing pigment epithelial cells. The second study is out of the Riken Center for Developmental Biology in Kobe, Japan, and they're treating wet macular degeneration. They're trying to treat wet macular degeneration. So in the advanced cell therapies approach, they isolate one of those cells from the 8-cell embryo. They grow it up, get a whole pile of them, either freeze them away or use those transcription factors to turn them into pigment epithelial cells, and then they freeze the pigment epithelial cells away and they store them as a slurry, as just a slurry of cells in the solution, and those they inject subretinally in patients with Stargardt's disease, 9 patients in that clinical trial, dry macular degeneration, 9 patients in that clinical trial. They've put in, they're divided into 3 groups of 3, and the groups get either 50,000 cells, 100,000 cells, or 150,000 cells. And to give you an idea how recent this is, the first paper, and it's the only paper that's been published on this that I've been able to read, came out on October the 15th, and this is out of the Advanced Cell Technology. Their study has shown, I've got to hope that this is going to work here. So what they've done in that trial is they took their cells, they injected them by needle under the retina, and they infused them in, and then they've given time. And the results so far have shown that the cells have engrafted. The cells are in there, but they formed clumps of pigment, and they haven't formed sheets of pigment. There has been some early indication of visual improvement, but that wasn't the purpose of the study. The purpose of the study was to show that it was safe, that people weren't going to develop tumors by putting these cells in the eye, that they weren't going to get other side effects. So, but despite that, the early results have shown out of the 9 patients from the dry macular degeneration, 4 of the 9 within 6 months had 15 letters of improvement in their vision. And in the Stargardt's disease, 3 of the 9 had up to 15 letters of improvement. So there has been some improvement. That wasn't the purpose of the study. The study that I'm more impressed with is the Riken study. And the reason I'm impressed with it is they've taken stem cells, which now gets away from the ethical and the moral issue of working with embryos. They've taken transcription factors, and they've cultured the skin cells with some, and it took time, and that's the hard work to find out which transcription factors, but 4 transcription factors would turn the normal skin fibroblast, a type of skin cell, into in what's called an induced pluripotent stem cell. And a pluripotent stem cell can form any cell in the body. And then they found, and that was 2 years ago, one of the researchers, Dr. Yamanaka, received the Nobel Prize for the work. So we're just about done here. They then reprogrammed those cells to become stem cells, and they did so with some simple organic chemicals, and cultured them up to form a mat of pigment epithelial cells. They then, that's the chemicals, they then took rectangles of those cells and implanted them. Now, what these look like for those that are not sighted, if you've ever felt or seen an aggregate concrete driveway where you've got all the pebbling in it, that's what this looks like to me. It almost looks like aggregate. And so these are pigmented cells, and they behave and function just like regular biologic stem cells do. They've taken those, And the first patient had surgery done on the 12th of September this year. And they're doing 6 patients altogether. They went in and they took out the epiretinal membrane. So they took out the blood vessels, they took out the scar tissue, and they took out the disrupted pigment epithelial cells that were there. They've implanted the sheet of pigment epithelial cells that they generated from scratch, and that's where it sits right now. We don't know what the result is because there hasn't been anything reported, but those 2 are the only 2 trials that I'm absolutely aware are underway. There are pros and cons, but I will address those in question if there are any.
Yeah, my question is, would that stem cells work on a person who has like tunnel vision?
So with retinitis pigmentosa, which is what you're talking about, there is a degeneration of the photoreceptor cells. And this technique is not putting in photoreceptor cells, it's putting in the pigment epithelial cells. But also, from that formulation they use, they also have been able to generate photoreceptor cells. But it's a much more complicated integration to get the photoreceptor cells to hook in to the rest of the retina. That's a step that's coming.
How long has the process taken to date? How long have they been working on this? Would you estimate? Yeah.
The work has really been going on, since Watson and Crick figured out the DNA in 1953. So it's been a slow process. This is really speeding up. It takes about a year by the time they extract the cells from the arm, grow them up into pluripotent stem cells, to transform them into retinal pigment epithelial cells, and they've just got the approval to do the study. So it would probably take about a year based on this technology, but it's going to evolve.
I can hear the excitement in your voice.
Yeah, well, it is exciting.
And I know it's exciting for all of us probably that are interested in this thing, and I know it's a big question, and technology gallops along as technology improves and the stem cell research and stuff like that, but is it going to be 2 years, 5 years, 10 years, 25 years?
Okay.
What do you think?
It's not going. It's going to be way before 25 years. 10 years is the window that I'm thinking of in terms of. But it's in that 10 to 20 year time frame. I'm an optimist, so I'm thinking that it's likely going to be 10 years. But with this kind of innovation, it can turn on a dime. Like this has really, turned on a dime. What's come out of Japan is absolutely amazing. But, as I said, one of the members of that lab, committed suicide because of the hubris that's in there. Any other questions? Question?
Yes. Is there any research being done as far as stem cells and the optic nerve?
There is. I haven't gone into that literature in great depth. What we do know is that taking these pluripotent stem cells, culturing them in the presence of vitamin A, retinoic acid, and in one of these transcription factors called sonic hedgehog, will turn those pluripotent stem cells into nerve cells with axons and the branching point. So what's important is to find to get the cookbook recipe, to get the recipe that will turn that specifically into a ganglion cell. And that's what's going on right now, is people are working around the world to figure out the recipes. When we started growing wheat 10 or 12,000 years ago, we didn't get a perfect loaf of bread right away. It probably took about 1,000 years to get a reasonable loaf of bread. We're going to do that far faster.
And so, I know there's some of these clinics in Mexico talking about, you can come down and they'll give you
That's what this talk is all about.
That is the hype.
I'm going to give you a very specific example of one of my own patients who has an inherited disease that is lethal. Yeah. Inherited disease that is lethal. And she said, I'm going to go down to Costa Rica because I can get stem cell transplant down there. And I told her it's not going to do you any good at all. It's going to cost her $30,000. She mortgaged her house and she did it anyway. And I told her ahead of time, I said, at the very best, what's going to happen, they're going to put those stem cells into your body. Your body immune system is going to recognize it as foreign, and they're going to have a $30,000 lunch. That's what I told her. I said, at worst, what's going to happen is you're going to get those stem cells in your body, and they're going to have a mind of their own, and they're because they're unregulated. They don't have all of those, like the marionette isn't there tugging on them, the cilia isn't there telling them what to do, the chemicals aren't there telling them what to do, and they can become cancer cells. And so that's a big risk with it. So that's the big risk. So those places down in Mexico, they're not going to do you any good. You want places like Riken, you want places like Advanced Cell Technology, you want places like that, and I wouldn't be the first in line to get this therapy because you don't want to put all your eggs in that basket. You want to wait for a little while until the technology matures. But it's there, and I'm hoping against hope. I'm going to live long enough to be able to see that come to fruition. Yeah.
We have time for one more question.
So while that's coming, one of the things is there that with the embryonic stem cells, they're not the person's own cells, so their own immune system runs the risk of knocking those cells out. And I've looked after patients for years that have had stem cell transplant for leukemia, and I'm not looking at it from a leukemia perspective, but from an eye perspective. And I've also looked after patients for years that have had solid organ transplants, liver transplant, kidney transplant, pancreas transplant, all those heart transplants, lungs. All of those people are heavily immunosuppressed so that the patient's own body doesn't turf the transplant out. And the risk of the immunosuppression is increased risk of diabetes, increased risk of cancer. There's all sorts of side effects there, and it requires supervision. So the fly-by-night places that says, yes, I'm happy to give you for $30,000 a shot of stem cells, you say, no way. Leave them alone, absolutely. And there's a question over here.
Yes, this is more so for inherited genetic diseases, like for people with retinitis pigmentosa. Hypothetically, let's say you're able to heal the retina, because as far as I understand it, it was the X chromosome that was defective.
In retinitis pigmentosa, there are hundreds of different genes that have been implicated because the cells that are involved, primarily the photoreceptor cells, but the pigment epithelial cells as well, each are run by 2,000, 3,000, 4,000 different genes. And any one of those genes potentially could be defective and result in retinitis pigmentosa. So you talked about the X chromosome.
Yeah.
That really only affects the males. So males will get a single RP gene and develop the problem. And then there's dominant forms and recessive forms. So right now, I don't see immediate benefit for it, but if we can go in and regenerate a retina and the photoreceptor cells and the pigment epithelium in these macular degenerative diseases, then there's no reason why we couldn't go in, get the cells out of the retina that are there and defective in RP and put in ones that would work better. But it means swapping the genes out that are causing the problem in the first place, because if we put the patient's own cells back in, they're already pre-programmed for that problem. Does that make some sense?
Yeah.
No.
My question was just that, even if you could, even if it was replaced surgically or healed with stem cells, is there any possibility that it could continue to deteriorate because it was more like you fixed the symptom but not the cause?
Oh, absolutely. Look, so if you've got, if the stem cell carries the abnormal gene, which is just what I was saying,
yeah.
you've taken out the tissue that's now defective, but you replaced it with cells that are going to make more defective tissue. So you have to swap the gene out as well as putting new cells in. So that's 25 years away.
Okay, thank you.
Okay. Hi, I'm sorry, I have to leave it at that. I want to thank Dr. Wyse for sharing his vast source of knowledge with us. So let's give him a big round of applause.